Okay, then I want to present to you Orit from Israel.
Thank you. Wait, let me see this. Okay. Hello, everyone. My name is Orit, and I'm from Israel. And I did this project during my high school years in the Weizmann Institute of Science. And my research focus is link RNAs, which is a type of non-coding RNA, and its effect on neuronal differentiation. And worry not, I will explain that now. So, as some of you may know, the central dogma of molecular biology states that we go from DNA to RNA to protein, but sometimes it's not like that, actually. Sometimes there are RNA sequences that are not translated to protein, and they just stay in protein shape. So we know of some non-coding RNAs that have function, but there are still many that we don't know much about yet. And when non-coding RNA is longer than 200 bases, we call it long non-coding RNA. And inside this group called long non-coding RNA, there are some that do not overlap with any other genes. This allows us to study them without a complication caused by overlapping. These are called long intervening non-coding RNAs, and this is what I was studying. So since long non-coding RNAs have very high expression in the nervous system and during processes of cell differentiation, which is the change from one cell type to another, and cell development, I wanted to see if they have any effect on these processes and if they are important in the nervous system. So for this reason, I chose a cell line called SHSY5Y, which can easily be prompted to either differentiate into a more neuronal state or keep dividing. So I chose 3 genes which are long non-coding RNAs, SOX9AS1, PVT1, and Cyrano. And I wanted to see what would happen if their expression in this differentiation process was changed. So I wanted to see what would happen if first I silenced the genes, which means causing them to not appear at all during the process, and what would happen if I overexpress them, which means causing them to appear too much and unnaturally. So for silencing, I used a method called Rna Interference. In this, we use sequences of double-stranded RNA, which usually only appears in viruses. So the cells are going to recognize that as viral, and they want to destroy it and get rid of it. So we disguise our long non-coding RNA as double-stranded RNA. We put it into the cells, and they are going to get rid of it for us. And whenever they see a matching sequence, they are going to destroy it as well. So this way, we get rid of the expression. And here, we have plasmids. This is for overexpression. And plasmids are circular molecules of DNA, which usually appear in bacteria. We put our sequence that we want to overexpress into the plasmid and put the plasmid into the cell. When a cell has a plasmid inside of it, it knows how to transcribe information out of it. So it will transcribe the gene that we want and cause its expression to rise. That was too late. So for 2 of the 3 genes that I checked, I didn't have clear results, and I couldn't really tell what was the answer. But for one of the genes, SOX9AS1, I had very clear and interesting ones. So as you can see here, the cell number in the culture in which I silenced the gene, this one, is much higher. This means that the cells tended to keep dividing instead of differentiate. You can even see this very clear in the pictures. In the silence culture, it's much more crowded, there are much more cells. And you can also see over here that they are shorter. This means that they didn't grow longer neurites and get into this more developed stage of neurons. So this is very interesting because it relates to a previous study about the nearby protein coding gene SOX9. SOX9 has the opposite effect on these cells. It causes them to keep dividing instead of differentiating. So this hints that perhaps SOX9AS1, the non-coding one, functions by regulating the expression of the nearby protein coding gene SOX9. And this basically opens us a doorway to further research on long non-coding RNAs as regulators of gene expression. Questions?
Thank you very much. Do we have any question? Up there, yeah.
Yes.
what was the reason for you choosing those 3 genes? Like,
Oh,
was there a
yeah,
reason
there
behind
is
it?
a reason. There are several reasons. We had several criteria when we chose from like many non-coding RNAs, and for example, we wanted to use genes that have very high conservation between far species. So like if I have a gene that expresses both in humans and in like zebrafish, that means that it's probably important for something. Otherwise, it wouldn't have stayed for so long. And we also wanted genes that have high expression in the brain and in the nervous system, so we checked that in mice. And we wanted also genes that express highly during the process of differentiation. So for that, we checked the process that happens naturally, and we chose genes that in this process, they have high expression or have expression that like rises and falls. So that indicates that perhaps they're important for the process.
Thanks.
Are there any other questions? Over there. Yeah.
How were you able to study something so intricate in high school?
I was actually very lucky. I got into a program in Israel which is called the Alpha program. And it's a program in which high schoolers work together with PhD students who are their advisors. So I got an advisor whose field is non-coding RNAs, and she helped me choose this topic. And she was like guiding me through the whole process. So I really wasn't alone, and I got a lot of help.
Okay, thank you.
No problem.
Any other question? Yeah, down here.
Yeah.
What was your method for researching this? Like, did you use cells that you had obtained and grew, or how did you?
Yes, the cells, we aren't making them ourselves. They are like, this is a cell line that has been studied many times before, and it's used very often for research of neuronal differentiation and neurons in general. And there is like a recipe for how to make them go through this differentiation process. So it like says 4 days in this kind of serum, and then you change and do that. So it's like the cells themselves have a method that is already defined by other researchers. So it's very simple.
Okay, thank you. Any other question? Or, yeah, one more question down here. Can you raise your arm again? Okay, thank you.
Hello. I couldn't recall. What is the purpose of this? What can you do with this knowledge that you have come to?
Okay, so I get asked that a lot. So first of all, I'm gonna say that it's important to know about everything that happens in our body. And this topic of non-coding RNAs is really new, and we don't know much about that yet. And this is pretty crazy in my opinion, because there are so many non-coding RNAs that we don't really know what they do yet. And I think it's important to just figure out how do they function and what is their purpose. So if we really find out that many long non-coding RNAs function as regulators of other genes, this can really help us in the future. And it can tell us more, for example, about non-coding RNAs in general. And it can tell us about inherited problems and what happens when these genes go wrong. It can help us solve these problems. And yeah.
Good work. Do you think it will work in the end?
I actually left this lab already, but everyone in it is working on long non-coding RNAs, and they're doing like amazing research over there, and yeah, it's going in a good direction.
Very good. How long time have you put on doing this?
I was working on this for around a year, but most of it was like writing the research, studying, and preparing for the actual part in the lab.
Very nice. Good work. Thank you.
Thank you.
Thank you very much.
Thanks, everyone. Have fun. Do science. It's fun.
Great, thank you.